This is video 5 for chapter 3, a review of biology for anatomy and physiology. So in this video, we're going to give an overview of cell organelles using this diagram of a typical animal cell. You do not need to know the functions in depth for these. So we're going to come up with very short functions for these different organelles. So if we look at these, we're going to skip a couple of them, but I'm just going to start over here with letter A. So if we look, A is pointing to this structure on the outside, and we've already covered this. This is the cell membrane. And if we were to summarize what the cell membrane does, it controls in and out. So it controls things going in and out of the cell in a nutshell. We are going to skip letter B because that is an actual function, and we'll mention that here in a little bit. But if we go to structure C is the nuclear envelope or envelope, or the nuclear membrane. Either one works for me, but this controls in and out of the nucleus. And that gives us letter D, which is the nucleus. The nucleus is usually the control center of the cell because it contains DNA. So this has our instructions for the cell, for its structure and its function. So it houses that. That's why it has its own membrane, because we need to protect that, so we control what comes in and out. E is a structure within the nucleus called the nucleolus or nucleolus. I've heard it pronounced both ways. Doesn't matter to me how you pronounce it. But this helps make RNA and assemble ribosomes. Letter F, which is all of this substance here. It's kind of hard to see the arrow going in there, but letter F is the cytoplasm. Sometimes it's referred to as the cytosol. We also said that there's intracellular fluid in there. So this suspends organelles. So the organelles, some of them are freely moving throughout the cytoplasm. And it acts as a site for reactions. So if something is releasing enzymes into the cytoplasm, reactions can take place in there. So we might have enzymes located in there that do certain things. If we look at letter G, it's this structure right here. Letter G and letter G prime, which is this one right here, are the same type of organelle, but we have two different versions of it. So letter G is smooth endoplasmic reticulum, which we can abbreviate ER, just to make it easier. This smooth ER makes some lipids and carbs. So it manufactures, it's like a little factory. G prime is rough endoplasmic reticulum, or rough ER. And you can see that it looks like it has a case of measles. It has little dots all over it. Those little dots are going to be ribosomes. So it's an attachment for ribosomes. And we're going to discuss a pathway here in a second, but an attachment for ribosomes, and it modifies proteins. H are the ribosomes itself. So some of them are on the ER, and some of them are free floating in the cytoplasm. So you can see both in the diagram right up here. You can see these little spots, and then we have some attached. So they can be in both places. Ribosomes are pretty important in our body. They are responsible for protein synthesis. So these are the sites where proteins are made, and that's their primary function, is making protein. I, which kind of looks like these flattened sacs with little bubbles on the end. This has a funky name also. This is called the Golgi apparatus or Golgi complex. This modifies and packages proteins. Letter J. Letter J kind of looks like a kidney bean with a bunch of squiggles inside of it. These are the mitochondria, and that's a plural. These are responsible for making ATP. So these are the powerhouses of the cell. So cells that have a lot of mitochondria are ones that make a lot of energy, a lot of ATP, but they also might be cells that need a lot of energy. So muscle cells have a lot of mitochondria in it because we use ATP to move our muscles. And these have a very interesting theory on how they formed because they have their own DNA. And it is handed down through your maternal lineage, so through your mother's DNA. And it is thought at one time that they were individual cells and they were absorbed by other cells and became a symbiotic relationship where they both benefited. So they're thought to have like bacterial origins. It's a really interesting theory. Letter L. And I'll move this up so you can see it a little bit better. Letter L, lysosomes. Lysosomes digest things. So they have a pretty tough membrane around it. And you can see that they have lots of little dots up in the diagram. Those are digestive enzymes. So if you need to break something down, like an old organelle that's no longer functioning well, the lysosomes will attach to it and begin to digest it, and then we can recycle those parts. Whenever a cell dies, the lysosome membrane can no longer be maintained, and the lysosomes break open, and the digestive enzymes just start digesting the cell, and it eventually will die. So there is some programmed cell death that's caused by lysosomes, and that's part of the decaying process. When a person or an animal dies, the lysosomes will eventually break open and the cells will start decaying, and that is natural process of death. Letter K. Letter K kind of looks like a lysosome, but you'll notice that there are no dots in the inside. This is called a peroxisome. And these break down hydrogen peroxide. H2O2 is hydrogen peroxide. Hydrogen peroxide is toxic to living cells, and it is a byproduct of some reactions. So we have organelles that will take that and they will break it down into water and oxygen, which we can use. Letter M, and I kind of did those out of order, so it should be, oh well, K then L, but that's okay. Letter M stands for the centrioles. And centrioles are involved in processes that we'll talk about later, but these make spindle fibers for mitosis and meiosis, which are processes of cell reproduction. And then if we look at letter N. Letter N above, we have these two little strips. These represent the cytoskeleton. And a cytoskeleton is internal structure. So we have different protein rods that act to give the cell a little bit of structure, but they can be made and broken down very quickly by a cell if the cell has to move. So I said that we would talk a little bit about these arrows here. So we have an arrow coming out that's representing exocytosis and arrows coming in that represent endocytosis. So if we imagine that this is a balloon and we were able to poke a hole into, put our finger into the balloon, it starts, the balloon would actually fill in around our finger. And that's what happens during endocytosis. And eventually this kind of like bubble that forms on the inside, it will seal back up, and anything that we want to get into the cell in bulk will be surrounded by cell membrane. So part of the cell membrane forms a bubble around it. Exocytosis, these little bubbles here that form from the Golgi actually will travel and meet up. They are made of phospholipids around here. They will travel through the cytoplasm to the cell membrane and merge with it, like over here, and it will either have a layer of phospholipids that surround it that can travel, or it will break open right here and release its substances in. So with that, we actually have, we'll talk about this during protein synthesis, but we'll come back to this diagram and talk about a pathway from the nucleus to the endoplasmic reticulum to the Golgi to the outside. The only other thing that I want to mention with cells is I'm going to draw a little diagram here. So if we have a cell, when we have cell movement, we can have different ways that it moves. So little hair-like structures on the outside that can move back and forth and propel the cell are called cilia. But we can also have a very long structure called a flagellum. Flagella is plural. Sperm cells have a very large flagellum that help propel it. So it's like having a tail that they wag back and forth very quickly, and it causes them to be propelled forward. So either little hairs all beating in the same direction, or one large tail.